Professor Zhao from Zunyi Medical University, China.
Hi.
And the topic is microRNA7 and NSCLC. Thank you.
Hello, everyone. Can you hear me? Good afternoon. Nice to see you. I'm so honored to share the topic microRNA7 and non-small cell lung cancer. As an important regulator in various biological process, mRNAs, small non-coding RNAs, with a length of about 22 nucleotides and encoded by corresponding genes. Studies have shown that the abnormal expression of mRNA is closely related to tumor regenesis of many kinds of tumors. Which suggested that it might represent a new class of targets for the development of novel therapeutic strategies against the tumor. MicroRNA7, termed as MIR7, used has a high degree of conserved mature sequences in different species. The biology of MIR7 is complex as it has important functions in normal cellular development as well as in several disease states. And interestingly, recent evidence has shown that MIR7 plays an important role in growth, migration, and invasion of multiple cancer, indicating that it may be a new target for the diagnosis and treatment of these cancers. In our previous work, we reviewed that the CBPR MIR7 axis controls CD4 positive T cell activation and function and orchestrates the pathology of experimental autoimmune hepatitis. In addition, we also found that MIR7 could regulate other immune-related cells, such as macrophage and neurons, and contribute to this related disease, such as the acute lung injury. Briefly speaking, in our series work, we revealed that MIR7 might be a novel promising regulator in immune reaction-related diseases. According to the latest estimate of the global burden released by IARC on December the 15th, 2020, it inhibits that lung cancer is a still leading cause of death around the world. So, regretfully, due to multi-factor mechanism regulation, molecular regulation mechanisms, the efficacy and application of MIR7 is still need to be further improved. Based on our series work on the biological function MIR7 in immune-related disease and the non-negligible role of mRNAs in virus tumors, we want to explore more details about the relationship about MIR7 and NSCLC. The existing clinical data shows that the expression of MIR7 in the expression level of MIR7 is significantly lower in NSCLC tissues than the adjacent tissues, and which is associated with worse prognosis in patients with NSCLC. And so we want to know the potential mechanisms in view or in consideration of the fact that promoter mutation is one of the important factor affecting the expression of microRNAs moleculars. So we investigated the we investigate the integrity of Mir7 the promoter in lung cancer tissue using DNA sequencing. As shown in the figure A, we found that there were two sites, the mutation as specific sites mutations in Mir7 promoter sequences from cancer tissues. In figure B, we further found that mutations at specific sites could decrease the activity of Mir7 promoter and alter the expression of Mir7. Notably, mutations at these sites of Mir7 promoter were found to be closely associated with the poor prognosis of lung cancer patients, indicating that the integrity of promoter is an important facet for abnormal expression of Mir7 in NSCLC. Next, we confer the specific biological function of Mir7 in an SLC. And based on a series of in vitro and in vivo experimental settings, such as the tumor-bearing mice model, we found that Mir7 can inhibit the growth and metastasis of human NSCLC cells. Which is related to the altered transduction of AKT pathway. In a nutshell, Mir7 may license the tumorigenesis of NSCLC through controlling the growth and metastasis of tumor cells. Cancer gene therapy is a promising new technique for treating many types of cancers. Moreover, promoter-driven targeting gene therapy is an important field of cancer gene therapy. One obstacle of the application of this strategy in cancer is how to select an ideal gene promoter. TTF1 called serial transcription factor 1 as a lineage-specific oncogene is dominantly expressed in lung cancer, but not other types of cancer. And its expression is closely correlation with the prognosis of lung cancer patients. In order to investigate the TTF1 application value in the Mir7-based gene therapy in NSCLC, we constructed the TTF1 promoter-operated Mir7 expression chaotic expression vector. We terminate PT-Mir7 and analyzed the target efficiency and safety of TTF1 promoter-driven Mir7 expression in a nude mice model of NSCLC using remote injection with, as shown here. Expectedly, compared with control group, TTF1 promoter-driven Mir7 expression could disturb the growth of tumor cells in vivo, which was similar to our previous data. Interestingly, we detected the distribution of the plasmid PT-Mir7 in different tissues and organs, and found that the plasmid dominantly enriched in tumor and lung tissues. Notably, the level of Mir7 expression was significantly increased in tumor
Excuse me, ma'am.
tissue.
Sorry for the interruption. Ma'am, actually, you have only 12 minutes.
Okay. Sorry, Chairman, I'm not very here clearly.
You have only 12 minutes, so conclude it quickly.
Okay, thank you. So This data indicates that TTF1 promoter might be a much valuable candidate to improve the targeted expression of distinct gene in NSCLC cells, and is used to our following research working Mir7-based gene therapy against NSCLC. Finally, I would like to thank our group for contributing to this project. This is my supervisor, the Professor Xu and Xiong. And this is my classmates, and I'm very grateful for them to this subject. Okay, thank you for your attention and your time.